Okay, so warm good morning to all of you. Okay, welcome to the first session of the day. Okay, and it will be taken again by Dr. Rakti Mukherjee on the topic bits and pieces of developmental biology. Okay, so I hope you all will have a fruitful session today. So I hand over the session to Dr. Rakdim.
Thank you very much, Dr. Elizabeth, for introduction. Students, what I have decided is not to go with any specific topic because my statistical analysis with all these years being in with GSBTM suggests the majority of the lot is from microbiology and biotechnology background or even biochemistry, and very few universities do have developmental biology either as their basic or elective subjects. Of course, for those from animal sciences, I need not mention, it's always there. So this talk is basically to draw you towards this subject so that rather than leaving it as an option, you can choose this very important area where lots of questions are asked every year in CSIR-UGC-NET. And accordingly, I have given title for this talk as Bits and Pieces of Developmental Biology, suggesting I'll be covering the basics of several different areas. Of course, on and off, I will be conducting in different sessions all across Gujarat. So maybe next time you can catch me in entire exhaustive details of fertilization or any other topic. But right now, I'll be dealing with the bits and pieces of developmental biology. So when you talk about the developmental biology in general, so I'll be dealing with some of the basic concepts right now. And before I begin, I would like to start with some interesting questions. How does the body form its head always, and you have the head always above its shoulders? Okay, that's the first question. How come the heart is on the left side of the body? It's not on the right. Of course, there are a very rare section of individuals who are born with the right-sided heart. We call it as the dextrocardia. Again, how does a simple tube becomes a complex structure of the brain and spinal cord that generate both thought as well as movement? Why can't we grow back the new limbs? How does the sexes develop their different anatomies? Why everything is not common? Okay. Coming to development of a multicellular organism begins with a single cell, the fertilized egg or zygote, which divides mitotically to produce the cells of the body. Just give me a second for the basic setup from my end. Just a second, everything is fine. Okay. So what exactly is development? Development of a multicellular organism begins with a single cell, the fertilized egg or zygote, and which divides mitotically to produce all cells of the body. Okay. So therefore, this is very fascinating, that how a single cell gives rise to the entire organism per se. And that takes place because of the mitotic high rate of mitosis. The study of animal development has traditionally been called embryology, from the stage of an organism that it exists before fertilization and birth. And there is a lots of misconception. Many people call developmental biology and embryology together as a common term. But then why this subject is called as developmental biology, that is very important to understand. So we say, according to embryology, there's a stage of an organism that exists before fertilization and birth. This suggests that birth is a vantage point or a full stop where the development should stop. But is it worthwhile to talk that development does stop at birth? Okay, but actually, the development does not stop at birth. We replace more than a gram of skin cells every day. Just now, today morning, we all might have taken bath. Several skin have been sloughed off from our body. Again, we replace RBCs every minute of our lives. Even if we don't donate blood, as such, the RBCs are going to die in 180 days. So therefore, development doesn't stop at birth. And finally, even after we are born, the regeneration takes place. Not that prominent in humans, I will touch upon it, but of course in the amphibians, the newts, the salamanders, regeneration is quite apt. So these all brings us to the contention that developmental biology is an important area, and it encompasses all other disciplines of biological sciences. Some of them you might be already covering in this crash workshop, such as molecular biology, physiology, cell biology, anatomy, evolution and ecology, immunology, neurobiology, as well as cancer research. So it's a modern area, it's a modern subject, much more modern in the from the perspective of embryology. And in fact, to understand the key functioning of developmental paradigms, we need lots of support from cell biology and molecular biology. When we talk about developmental biology, there are two major questions that we always ask ever since we understand life. The first being, how does the fertilized egg give rise to the adult body? The second, how does that adult body produce yet another body? These are the two fundamental questions that are often asked as a part of developmental biology. In addition to these two fundamental questions, we always have 6 general questions that are scrutinized. And those 6 general questions, one by one, I shall talk. The question of differentiation, question of morphogenesis, the question of growth, reproduction, evolution, environmental integration. So along with the 2 specialized questions, we are supposed to answer 6 general questions that the subjects carry. At this point, I would like to tell to the students that the terms and terminologies of developmental biology itself features some 50% of the questions in CSIR-NET. Another 50% would go with your depth of understanding of a particular topic. So currently in today's talk, I am just dealing with the 50% of the terms and terminologies, and I will give live examples in the form of questions which will enable you that yes, with certain terms and terminologies, how questions are framed in CSIR-NET. Of course, now it is CSIR-NT-NET, that would be more. National Testing Agency is the one who conducts, but still we use the old term. So we turn to growth right now, and there are questions asked from growth. And the first part I would like to highlight is the difference between isometric and allometric growth. What's the difference between isometric and allometric growth? Anyone who is already aware can just give some cue, and then I will proceed ahead. Okay, isometric versus allometric growth. First, we will talk about isometric growth. There are many animals which undergo isometric growth, and those they mature from new hatchling to adult. While they mature from new hatchling to adult, all the body parts go at approximately the same rate. So there is a symmetrical body growth. At the same rate, the entire body growth takes place. And the adult proportions are not significantly different from that of the juvenile. Some of the common examples that I would like to give is the human heart and the salamander. So here I have represented the salamander. If you can see the newly hatched, the newly hatched salamander and the salamander adult, you can see that there is a close resemblance. It seems as if the adult animal is just the extended or a zoom version of the newly hatched one, or likewise the vice versa. The newly hatched organism is miniature version of the adult. And how it is possible? It's a symmetrical growth of all the organs. Okay, say for example, organ X, the growth curve is similar. Organ Y, the growth curve is similar. Likewise for Z, so on and so forth. So some of the examples are human heart. Right from the, if you see the child's heart, then middle age, adult, the juvenile, you can see that there is a symmetry in the way the heart grows. Likewise, even for salamander, just now it's projected right in front of you. These are the examples of isometric growth. So let me summarize. In isometric growth, you have all body parts which grow at approximately the same rate, and the adult proportions are not different from that of the juvenile. So that's the isometric growth. Now I will switch over to the next term, that is allometric growth. And I think rather than my explanation, it's always said in English as per the proverb, a picture is worth 1,000 words. So the diagram says it all. Allometric growth, in a species that exhibits allometric growth, the different cell lines or body parts grow at different rates. That was a symmetrical growth at a same rate. Here, the different body parts grow at different rates. And when I say rate is always not a unified parameter. It is always a comparative parameter. So here, the comparison is in the context that it is relative to an ancestral isometrically growing form. So that's the development that takes place. So accordingly, you have the best example is humans, right? When you talk about the humans, the humans are the best examples of allometric growth, the head, the limbs, the body grow at different rates, resulting in human adult with proportions. This term with proportion is very important. Two examples I would give. One from developmental biology. Of course, you can see when we are born, any newborn child, if you see the infant, the head is considerably symmetrical. It's not perfect symmetry. The head is larger as proportion to the body. Okay. But right from this infant, as you compare that with adult, here the symmetry is formed. The remaining body parts considerably grow, and then some form of symmetry is maintained. But then with respect to each organ, there is asymmetric growth, unlike isometric growth. So there is asymmetric or allometric growth. Examples are we humans, we grow in the form of allometric growth. Now, whatever I talked so far, that is in the context of growth, I will give some example that simply from this 15 to 17 minutes of talk, what all questions can be asked. And the next one will be from growth. And basically, the question will be based on the concept of growth, how you have understood what is meant by growth from the context of developmental biology. So here I shall project the question. In developmental biology, what is meant by concept of growth? Option A, growth occurs through increase in cell size. Option B, growth occurs through increase in cell number. Option C, growth can result from increase in volume of extracellular matrix or ECM between cells. And option D, all of the above. What would be your correct answer? Either you can interact or you can go live in your YouTube chat box. I have already opened YouTube in one of my windows, so I will be very much. Okay, Nirali Tandel has shot D. Let me see others what they are answering. Hardy Shah is going with D. Vrinda Patel with D. In short, majority of the students are going with D. I don't want to go with KBC style that. Likewise, even in Zoom also, Zoom chat box also, people are going with option D. Some of them are Neha Patel, Hina Vidhani, Rajesh Kataria, and Ravi Trivedi. Most of them are going with D. And yes, most of you have guessed correctly. So should I say this is the beginning of your preparation for clearing entrance? The option D is correct. All such features are true in context of growth, that during growth, there's an increase in cell size, increase in cell number. There is an increase in volume of extracellular matrix between the cells. So all the options hold true from the context of growth. Another question from growth. This time, this question which I shall be talking is an integration of the developmental biology with cell biology. Some of the faculties might talk about cell cycle and its regulation. So that context you have to get, only then you can answer the next question. So my suggestion to the student is don't put compartments in between the subjects. In fact, it's a holistic life science. You cannot create compartment. Every subject is related to another within the life science discipline. And not only life science, even physics, mathematics, and chemistry also are a part of it. So what's the next one? Many cells in the body divide only rarely, if at all. Neurons, red blood cells, keratinocytes are extreme examples. The question is, in which proportion of the cell cycle or which portion of the cell cycle would such cells be considered to be? Where would you find those cells, whether it is in G1 phase, G0 phase, S phase, or G2 phase. You can hit the chat box either in Zoom or YouTube Live. Well, both chat boxes are going with the common consensus of option B. And in fact, those who have studied cell cycle in either this crash shop or as your subject, for them it's very easy. The correct answer, as you have guessed correctly, is option B, that's the G0 phase. So it's a portion of the cell cycle G0 phase where the cells would be when they divide only rarely, except for certain examples. Now I would come to the principles of development, and each principle followed by a brief overview of maybe a line each of the principles of development. Again, from the principles of development, many questions are asked. The first principle of development is differentiation, or how an egg becomes an adult. So one of the various ways by which an egg becomes an adult is differentiation. The concept of differentiation answers how do cells become different from one another and also from their precursors. How do cells become different from one another and also from the precursors? In very simple language, I would say differentiation refers to a less specialized cell becoming a more specialized cell. So once the cell becomes more specialized, obviously is going to be different from the precursors. The next principle of development is morphogenesis. Morphogenesis tells us how an ordered form is generated, why random events do not take place, how entirely ordered form is generated, that is morphogenesis. Likewise, the third principle of development is growth. Growth explains how size is regulated. Dear students, you all might be aware that in neoplastic condition or during cancer, there is an abnormal growth. Since I am talking about developmental biology, let me tell you, some of the aspects are very common with respect to cancer and regeneration. Some of the aspects are common. Even in regeneration also, high rate of mitosis is there. Even in cancer also, high rate of mitosis is there. But then in regeneration, during the repair process or regeneration, the high proliferative index might be there. But after that, the cell returns to the cell cycle. Okay, the cell returns to the cell cycle. So again, the maintenance of growth and the growth size is very important. That is growth. Reproduction. How does one generation create another generation? That is successive progeny, that is reproduction. And finally, you have evolution. Evolution answers the query, how do changes in developmental process create new anatomical structures? That is evolution. We all know that initial days giraffe was very short in height, but over a period of time, the trees became less. The leaves were hard to find, or it was at very height, it was considerably high from the ground, and therefore the adaptation took place. And now we have most of the giraffes which are considerably tall. So evolution answers the query, how do changes in developmental process create new anatomical structures? In addition, there are 6 general questions scrutinized: question of differentiation, morphogenesis, growth, reproduction, regeneration, evolution, and environmental integration. What is environmental integration? Can anyone tell me? What is environmental integration and why it's so important? Why we don't ignore? Otherwise, differentiation, morphogenesis, growth, reproduction, it could have been enough. What is environmental integration and what's the function of environmental integration? Environmental integration is the process by which you get along with the environment, you integrate with the environment. There is no backlash from nature. That is environmental integration. Now we come to the objectives accomplished by development. Some of the objectives accomplished by development are: the first one, it generates cellular diversity and order within the individual organisms. Second, it ensures continuity of life from one generation to next generation. This continuity of life is there from one generation to next generation. Those are some of the objectives accomplished by development. There are 2 fundamental questions of developmental biology. How does the fertilized egg give rise to adult body? And how does the adult body produce yet another body? We have already covered this. Further, a single cell that fertilizes the egg gives rise to hundreds of different cell types, such as some of the examples are the muscle cells, the epidermal cells, the neurons, the lens cells, the lymphocytes, the blood cells, and fat cells. The fat cells are adiposomes or adipocytes, the muscle cells. The generation of cellular diversity is called as differentiation. One of the ways by which you can define differentiation is the generation of cellular diversity is called as differentiation. I always tell a less specialized cell becomes a more specialized cell. Now we will raise the question of differentiation. Since every cell of the body, with few exceptions, contain the same set of genes, how can this identical set of genetic instructions provide different type of cells? That's the first one. Second, how can a single fertilized egg generate so many different egg types? Then the question of morphogenesis. The question of morphogenesis helps us to understand how can the cells in our body organize into functional structures. our differentiated cells are not randomly distributed. They are rather evenly distributed. And further, not only they are randomly distributed, they are, despite the fact they are randomly distributed, they are organized into intricate tissues as well as organs. They are divided into intricate tissues as well as organs. And once the development takes place, the cells divide. Once the cells divide, they migrate, and many of the cells would die, but there are others that undergoes a repair process. So development, the cells die, they migrate and die, and the tissues will fold, and after that it would separate. And based on that, we have, they are organized into intricate tissues and organs. During development, the cells divide, migrate, and die. Tissues will fold and separate. So that was about the morphogenesis. During development, the cells divide, migrate, and die, and the tissues will fold. And I think I will give you an example to help you to understand better. So when you talk about the question of morphogenesis, our fingers are always at the tips of our hands. Have you ever seen that the fingers are not on the tips of our hands? Is randomly, it has gone accidentally towards our feet or any other structure? That doesn't happen. Similarly, our fingers are never in the middle, it is always at the tip. Similarly, our eyes are always in our head. You won't find the eyes in your stomach or the gut or the toes. They are fixed structures. So this explains the question of morphogenesis. When we continue the concept of morphogenesis, it's because of morphogenesis that ordered form is created. And once the ordered form is created, it involves coordinating the cell growth, the cell migration, as well as the cell death or apoptosis. Again, can someone write in the chat box how many types of cell death are there? How many types of cell death are there? You can write in the chat box. One I already stated. How many types of cell death does exist? Devanshi Bhatt, your question, once functional, can the cell change its fate by any other mechanism? Yes. There are several other terms, Devanshi. You have to wait because I have organized my lecture in such a way that even before you ask, I think your doubts will be cleared. Just like earlier in Orkut and right now in Facebook, sometimes earlier someone writes their own photograph and later on they write committed. Committed means you are in relationship with someone, right? So likewise, even for the question of morphogenesis, or once functional, the cells can change its fate. There are terms such as determination, induction, right, and commitment. Once we come to those terms, you will understand, or rather your question would get answered. In the meanwhile, people have written apoptosis, necrosis. Okay, I would like to give you information that one more information I would like to give. There is a third mechanism also, which is a hybrid between apoptosis and necrosis. It is called as necroptosis. So that also you can go through after you brush up reaching at home. Okay. So cell migration and cell death is there, the question of morphogenesis. And again, answering the question of Devanshi Bhatt, once the cells are committed, it's very difficult that they will be reverting back. But of course, you have a term called as transdifferentiation. And though this itself would take some 15-20 minutes to explain, I would just give an example because you are all new to the subject, many of you, majority of you. Whenever a person suffers from diabetes, the problem lies in the islets of Langerhans, the beta cells of the islets of Langerhans. Of course, the SNS cells are always in plenty. There's no problem with the SNS cells. Few years before, in the NCCS, the National Center for Cell Sciences, Professor Bhonde and his team, Ramesh Bhonde, R.R. Bhonde and his team, what they suggested something called as transdifferentiation. What is meant by transdifferentiation? There are many examples. I am just giving one, that the SNS cells would transdifferentiate into islet cells. Either it can be induced, it's of course induced, that is not naturally, you're inducing it with growth factors or cytokines. So such transdifferentiation would take care of your diabetes, of course, right? I just gave an example to make things clear. Of course, it's a very vast subject, cannot justify in 2 hours. So Devanshi, we'll come to your further queries in a short while from now. When you talk about the question of growth, already had several questions related to growth from CSIR-NET. Further, when you talk about the question of growth, if each cell of our face were to go just one extra cell division, you would have been horrified. Just imagine, right? We always say that in the very first lecture of immunology, I said that science goes beyond super science. There is something who controls the master control gene of this universe. One single extra cell division, our face would have been horrible. We would not have recognized ourselves in mirror. So there comes the question of growth. How precisely growth is controlled. Further, each cell of our arms, very interesting. You try to understand that in a majority of the population, we don't find, right? We always commit mistake and afterwards we say sorry. Or we have just committed a mistake. But developmental biology or the creator doesn't do that, right? The arms underwent just one round of extra cell division. Okay. Suppose one extra cell division would have taken place. So the arms one of the arm would have been so huge in size or length that even without bending, we could have tied our shoelaces. Okay. But it's a magic or it's a phenomenon that both arms grow at a constant rate. Why only arms? Even your legs, the extremities, okay. So symmetry in growth is maintained. So that is a great blessing that unequal growth does not take place. Otherwise, we would not have gone with a fixed size in case of the females. The short, the medium, the long, the extra large, likewise the XL and double XL and L, it would not have taken place. One of our arm would have been XL, another would have been double XL. That doesn't happen. Okay. So that was the question of growth. Further, how do our cells know when to stop dividing? That's interesting, right? What is the grain? You all are driving the modern cars. Many of you are driving, right? You all might have heard about ABS system, anti-locking braking system, right? Or anti-skid braking system, ABS, computer controlled brake. The traction is not lost. exact braking or dead braking is possible without the car skidding. So likewise, students, have you ever thought, how do our cells know when to stop division? Right? Who is instructing them? Can now you stop dividing? That's interesting. And finally, one last, that how is cell division so tightly regulated? How cell division is so tightly regulated? Okay, so these are some of the questions of growth. So we just discussed isometric growth, allometric growth, which phase of the cell cycle the growth is maximal and minimal, right? So these are some of the questions when we discuss growth in perspective of developmental biology. Now comes the question of reproduction. The sperm and the egg are highly specialized cells, and only they can transmit the instruction for making an organism from one generation to the next generation. Now, interestingly, the question comes that should come in your mind: how are these germ cells set apart? And what are the instructions in the nucleus and cytoplasm that allow them to form the next generation? What instructions are given in the germ cell's nucleus and cytoplasm that allow them to form the next generation? So that's the basic question of reproduction. Next comes the question of regeneration. Regeneration, in fact, is a very vast topic, regeneration. Regeneration is the reactivation of development in post-embryonic life to restore the missing or the damaged tissues. That is regeneration. Interestingly, regeneration is a very important branch of developmental biology. We have a subject called as regenerative medicine, which takes care of the fundamental principles involved in regeneration, as well as some of the aspects that some tissues, especially in mammals, why the regeneration potential is less and what can be done to increase the regeneration potential. And regenerative medicine is further linked with tissue engineering. So these are some of the frontier areas of biological sciences. One of the potential benefit of harnessing the power of regeneration in humans would be that many people limb amputation takes place, and gangrene perfringes, anaerobic bacteria, or someone was in hurry to catch the train, the person has fallen off the platform, the limb is amputated. So with regeneration biology, the potential of harnessing the powers would mean that several limbs could be restored. No need for organ transplantation. Whatever organ is damaged, it can be removed from the body and it can be regrown. That fancy could have been done. Again, the nerve cells which get altered by age, by disease, by trauma could once again function normally. So there won't be any senescence, or you won't have any cerebral dementia that you forget. So neither, in fact, every male on this earth would remain as Salman, and every female on this earth would remain like Aishwarya. I just gave two examples. You can have your individual examples. The reason is that one of the reason is that regeneration would have solved majority of the problem. One more question has come from Pinkle Shah in the chat box. Cell death through activity by mitochondria. Yes, you are right, Pinkle. You have the intrinsic and extrinsic pathways. One is a mitochondrial death pathway, another doesn't involve the mitochondria. You are true. Right, maybe in some of the units that have also covered as a part of your CSIR syllabus. Now, when you talk about the question of regeneration, we all know that mammals are poor at regeneration. The mammals are poor at regeneration. While the mammals are generally poor at regeneration, there are some cells in our bodies, especially the stem cells, that are able to form new structures even in adults. Okay. Again, students, let me tell you, because it's a vast subject, there is something called a stemness. I explained in my immunology talk also when I was explaining about hematopoietic stem cells. You cannot allow all the available stem cells to differentiate. If all the stem cells differentiate to some other tissues or organs or some other cell types, you would not be left with available pool of cells. So some of the stem cells keep on regenerating, or they keep on maintaining the stemness. The mammals are generally poor at regeneration. There are some cells in our body, the stem cells, they are able to form structures. The question is, how do the stem cells retain the capacity, and can we harness it to cure the debilitating diseases? And that is the application part of stem cell biology. Okay. So I'm just naming it. Some of the experts from GSBTM would deal with this area. Dr. Digvijay Rana from Aribas exclusively talks on the stem cell biology and regeneration. Maybe sometimes you get opportunity, you can hear from him. This time, of course, he will be talking about the animal cell culture. So a very interesting way he teaches the stem cell biology. How do the stem cell retain this capacity, and can we harness it to cure debilitating diseases? The next diagram will explain the comparative regenerative capacities. So there you have Hydra, entire individual from small fragment. Okay. Planarians, whole anterior and posterior halves of the body. The amphibians and fishes, complete limb, tail, fin, fin regeneration in zebrafish. The lens, the CNS, heart, and other organs. So amphibians and fishes have good deal of regeneration potential. The mammals, of course, the regeneration potential decreases. So my dear students, if you can see, the base is broad, and as the regeneration proceeds, the tip gets narrowed down. Okay, suggesting that the mammals, the regeneration potential is less. The regeneration potential becomes less. Now we come to the area where many questions are asked: morpholexis and epimorphosis. And try to understand the term very nicely. Many questions are asked. At least I could find some 13 to 20 questions alone from this segment of my lecture, that is the last 20 minutes. Of course, I have taken for this presentation some 3 to 5 questions. Morpholexis and epimorphosis. Morpholexis is the regeneration of specific tissues in a variety of organisms due to loss of depth of the existing tissue. The word morpholexis comes from Greek word alloxene. Alloxene means to change. So morpholexis essentially means change. On the other hand, epimorphosis, it's a regeneration of a part of an organism simply by proliferation at the cut surface. So this was a complete organ, right? The regeneration would proceed by proliferation from this cut structure, that is epimorphosis. Things doesn't end over here. I have other explanations as well. So morpholexis and epimorphosis. Now let's see some of the steps involved in epimorphosis. One, of course, is amputation. the beginning, we start with either a natural or experimental amputation. Of course, lizard tail is a very important example. The salamander limb. So there you have the amputation. After amputation, there will be formation of blood clot. Several interplay of growth factors and cytokines does take place. You have the muscle myotube and the epithelial cells. Here the blood clot takes place. After the blood clot takes place, there is an event called as wound healing. It by itself is a separate developmental biology topic. Wound healing takes place, followed by dedifferentiation, blastema formation, redifferentiation, and morphogenesis. Some of the aspects of morphogenesis we just covered in our preceding part. This is how the steps are involved in epimorphosis. Once again, let's see the definition based on this example. Epimorphosis is the regeneration of a part of an organism by proliferation at the cut surface, by the proliferation of the cut surface. So therefore, here you see the cut surface, the proliferation takes place, and ultimately you end up with the complete structure. This is epimorphic regeneration. Further, when you go, this is a very important slide. Based on this slide, so many questions from CSIR-NET can be answered. Just understand, you have the types of regeneration. First, again, I'm revising. Epimorphosis: adult cells in some species de-differentiate into undifferentiated mass, and after that they again redifferentiate to form new structure. Example, regenerating amphibian limb, that is epimorphosis. The next is stem cell mediated regeneration. In stem cell mediated regeneration, the stem cells are the sources of lost tissues or organs. That is stem cell mediated regeneration. So the example of stem cell mediated regeneration is hair shaft regrows from stem cell in the hair bulb, as well as the replacement of blood cells from hematopoietic stem cells. Your hair fall is there, again new follicles and the entire hair grows. That's the stem cell mediated regeneration. Morpholexis, just now we covered, but again, it's the regeneration occurring through repatterning of existing tissue. And here there is little growth because repatterning takes place and there is very minimal growth, that is morpholexis. And last but not the least, we also have a compensatory regeneration, that the differentiated cells divide, but they maintain their differentiated functions, that is compensatory regeneration. Example of compensatory regeneration happens to be that of the mammalian liver. Morpholexis example is regeneration in hydra. Compensatory regeneration, that differentiated cells divide but maintain the differentiated functions. Example of compensatory regeneration is regeneration of mammalian liver. Now, I'm keeping this slide projected for just 20 seconds extra because based on this slide, so many questions are featured. And next slide will be again question answer session. Okay. So get ready with your chat box, some of the questions based on this slide. Just understand 4 terms and you can answer some 15 to 20 questions. Epimorphosis, stem cell mediated regeneration, morpholexis, and compensatory regeneration. There goes the first regeneration. First type. Morphalexis can be defined as, this from CSIR-NET December 2009. Option A, production of lost organisms by division in remaining cells, reinitiation of cell division in existing cells, followed by repatterning in those cells, production of complete organism by single cell, and movement of organism towards stimulus. Okay, those who are in Zoom, especially Rajesh Kataria, then Hina Vidhani, are going with option B. And those in YouTube, they are also going with option B. They are also going with option B, yes. Production of lost organism by division in the remaining cells. That, of course, is not the right answer. The option C, production of complete organism by a single cell, is totipotency. That is also not the correct answer. Plants, of course, are totipotent. Movement of organism towards stimulus is the taxis. Again, chemical movement is chemotaxis. So that's also not the answer. The correct answer, as most of you have rightly guessed, based on the definition that we studied, is option B, reinitiation of cell division in existing cell, followed by repatterning in those cells. So this gives you a very good idea that students, simply you learn and understand the definition, you can answer several questions from this subject. The next one, long one, important, CSIR-NET December 2011, right? Try to understand and only then you answer. Given below are some of the statements regarding regeneration. The type of regeneration characteristic of mammalian liver is considered as compensatory regeneration. Option. First fact. These are the facts, not the options. Fact 2. Regrowth of hair shaft from follicular cells exemplifies stem cell mediated regeneration. Third fact. Regeneration occurring through the repatterning of existing tissues with little new growth is known as morphalexis. Fact D, adult structures undergoing dedifferentiation forming blastema and then redifferentiate to form lost structure is called as epimorphosis. My question is, choose the most appropriate combination of correct statement. The first option, option D only, C and D, A, B, and C, and A, B, C, and D, that is all 4 options. Again, based on the single slide in which I included 4 definitions. Okay, let's see what students are answering. No answer as of now. Okay, it seems it's a difficult one. In fact, you need to interpret, only then you can answer. Many are going with D, ES. In fact, all statements regarding regeneration are from the previous slide where I projected the definitions. So the correct answer is option D, that is all four, A, B, C, and D is the correct answer. Okay, so that's about the type of questions these are asked. We move to the next one, again from one single slide, so many questions. This is based on the definition of epimorphosis, CSIR-NET 2013 December. Epimorphosis is regeneration through A, repatterning of existing cell that occurs in hydra, reinitiation of the division in existing cells followed by patterning as occurring in hydra, the reinitiation of division in existing cell followed by patterning as occurs in amphibians such as newts. Option D, repatterning of existing cells that occurs in amphibians. No thinking as of now. People are taking time just from one simple definition. No answer as of now. Nilesh, Agera, and Pinkle is going with option B. And those present from Zoom, they are going with option D as well as option C. It's a mixed response, in fact. Of course, without wasting much time, let me come to the correct answer. The correct answer is the reinitiation of division in existing cells followed by patterning, and this occurs in amphibians such as newts. And the correct answer is option C. Option C is the right answer, and that is what it fits into the definition of epimorphosis. Let's turn back. Okay, right. You have seen very clearly it is mentioned regenerating amphibian limb, right? So that's the answer. Yet another one from this segment suggesting that this portion is very important for your CSIR-NET developmental biology portion. This from if hydra is fragmented into various parts, separate group of cells repattern themselves into small hydra. Such a mode of development is termed as CSIR-NET June 2010. The four options are regeneration, morphalexis, epimorphogenesis, morphalexis, and epimorphogenesis. Which will be your right answer? Yes, this was a pretty simple question. Most of you are answering B. Yes, you are absolutely correct. The right answer is also option B, that is morphalexis. Maybe the last one for this segment before I go to the next segment. Okay, again from the similar area. In case of morpholectic regeneration, this from CSR December 2011, there is repatterning of the existing tissues with little new growth. Tissues after the stem cell division has taken place. There is a cell division of the differentiated cells which maintain their differentiated state to finally form a complete organism. There is a de-differentiation of the cells at the cut surface due to which becomes undifferentiated. These undifferentiated cells then divide to redifferentiate to form complete structure. What would be your answer? Rajvi, have you answered for this particular question? You can, if it's for this question, you can write yes in the chat box. Rajvi. Okay, Pinkal Shah, Seema Rana, everyone is going with. Even those in Zoom are also going with A, majority of them. And yes, if you can see, option A is the right answer. There is repatterning of the existing and tissues with little new growth. That's how the things unfold. Next, I move on to the next segment, the cycle of life. So that completes the second segment. Now I move on to the next segment, the cycle of life. We all know that the cycle of life is very important, a law of karmas or action of karmas. Here we would refer to cycle of life from developmental biology point of view. So students, for any animal or fungi or plants, the only way to get out from the egg to adult is to develop the embryo. Without development of embryo, the egg can never, the organism inside the egg can never get out to develop an embryo. Further, the embryo is where the genotype is translated into phenotype, where inherited genes are expressed to form the adult, that is called as the embryo. So, based on these two aspects, the developmental biologists usually find the transient stages leading up to the adult, and most interesting, the developmental biology also studies the building of organisms. It's also a science of progress and ever happening. The subject is quite rich. Now, what is the generalizable animal life cycle? The generalizable animal life cycle, one of the major triumphs of descriptive embryology is happens to be the modern animal developmental biology. It investigates the temporal changes in gene expression, that is time bound, and also the anatomical organization along this cell cycle, that is generalizable life cycle. We also have the generalizable life cycle So whichever organism you take, the organism has to pass through all these stages, and each stage is a separate topic in developmental biology. Fertilization, cleavage, gastrulation, oogenesis, the organogenesis, the birth, the metamorphosis, and finally, again, the gametogenesis. Okay. So every animal, whether it's a lower animal or a higher vertebrate, they have to pass through multiple stages of development. The stages of development between fertilization and hatching are collectively called as embryogenesis, whereas the fusion of gamete cells that stimulates the egg to begin development and initiates the new individual, egg activation, what you call. Then the subsequent fusion of gamete nuclei, you have the male and the female pronuclei, each of which has only half the number of chromosomes. Therefore, it's a characteristic for species, and this gives the embryo its own genome. Although fertilization is not under my correct ambit at the moment for this talk, but of course, since it is an important topic, I won't skip it altogether. Slight basics I will give of fertilization, and then of course, as and when further opportunity comes, we will talk about fertilization in detail. Right now, that the male and the female pronuclei, each of which has only half the number of chromosomes characteristic of the species, gives the embryo its genome. This is the summary of events. Actually, there are two models for fertilization in CSIR-NET syllabus. One, of course, is sea urchin, where Professor Eppel and his colleagues in 1978 started this research. And then you have the higher vertebrates such as mammals. In case of mammals, we generally so let's see the various steps. Let's see the various steps. In case of sea urchin, in the very first step, students, in fertilization, in case of a sea urchin, the sperm contacts the jelly layer of the egg. I know it's slightly difficult at this moment to understand because many of you might not be aware about the structure of the egg. Okay, so sperm contacts the jelly layer of the egg. The sperm has nucleus. The sperm neck has the mitochondria, which powers the sperm or provides energy. Remember, those sperms that have low motility or motility of the sperm is less, the mitochondria can be damaged. It can be seen, visually seen under electron microscope. And the acrosome reaction takes place. On the sperm head, there are acrosomal enzymes. These acrosomal enzymes will drill a hole across the jelly layer or extracellular coat. The fourth step is binding to vitelline envelope. This way, once it binds to the vitelline envelope, it moves its forward movement, and fusion of acrosomal process membrane and the egg membrane will ultimately lead to the fertilization steps. Again, I would like to give some new information that according to the latest research paper, sperms, in fact, earlier it was thought that the sperms actually swim through flagellar movement across the female reproductive tract, but now they have repeated the experiment using high-speed microscopy. That aspect no longer holds good. So fusion of acrosomal process membrane and the egg membrane that takes place. Once the two membranes fuse, then the further events will take place. Now, very important concept I would like to tell you. Okay, try to understand two aspects that I am telling you. One, organisms such as sea urchin the fertilization is going to be external. So both the male and the female would lay their sperm as well as the egg in the vast ocean. Again, there are different species of sea urchin present. Despite different species they are present, how is it that the acrosome reaction in sea urchin sperm, how it recognizes that it has to mate with only that species? Any answer? Well, dear, this is the area that I would like to show you. There you have the acrosomal enzymes, and after this acrosomal enzymes, you have these structures known as the bindin. It is the bindin which helps in recognition process. Now the question is, that was bindin. Very fundamental question. Why the sperms cannot move when they are in the testes of the male sea urchin, whereas the same sperm, when released in seawater, the pH elevates to 7.6. So why the sperms cannot move when they are in the testes? Should I give a clue? The first part is there is something called as pH. That is the first reason. And second reason is a cytoskeletal element, that is dynein ATPase. So just like the DDLJ movie we all have seen, probably Shahrukh was there. All throughout the movie, there was a dialogue that kahin pe nigahein, kahin pe nishana throughout the movie. So again here, the sperm attraction, that is action at a distance. Why sperm cannot move when they are in the testes? So I will give you the answer. The first reason is within the testes, the internal pH of is 7.2. Moreover, you also require lots of carbon dioxide because the gonads contain lots of carbon dioxide. So automatically when there is a carbon dioxide, strong base presence of carbon dioxide, the pH will become low. Okay. Further, the seawater, the pH elevated to 7.6, activation of ATPase leads to the sperm attracting at a distance. When you talk about the action at a distance, there are two proteins. One is resact, another is spiract. Both are from the sea urchin. Resact and spiract are 14 amino acid peptide. They are isolated from egg jelly of sea urchin. So we have a sperm plasma membrane. This sperm plasma membrane, you have the resac receptor. The resac will bind to the resac receptor. Once resac binds to the resac receptor, there will be conversion of GTP to cyclic GMP. GTP to cyclic GMP. Once GTP is converted to cyclic GMP, it will lead to the opening of calcium channel. The moment the calcium channel opens and there is influx of calcium, the influx of calcium would lead to the sperms which were non-motile would turn swimming, or it will start to beat its cilia and flagella. That's the role of resac. So what is the role of resac? Resac is sperm activating peptide. So resac as well as spirac. Whereas someone asks you what is the role of bindin, the role of bindin is species specific recognition. Remember these two areas, several questions are asked. Just to give you an idea, I will project few of them. So both these resac and spirac, they are 14 amino acid peptide, and these 14 amino acid peptide has been isolated from the egg jelly of sea urchin. Okay. Now let's have some question based on the background of last 10 to 15 minutes. Successful fertilization in sea urchin demands specific interaction between proteins and receptors of sperms and egg. In view of the above, which one of the following combination is correct? From CSIR-NET December 2016, binding in acrosomes and binding receptors on egg vitelline membrane, option A. Option B, binding in egg membrane and binding receptors in acrosomes. Option C, resac on egg jelly and binding on sperm membrane. Option D, proteasomes on egg membrane and complex sugar on sperm membrane. The correct answer, of course, no, I don't know for what reason. The correct answer is binding in acrosomes and binding receptors on egg vitelline membrane. My dear students, already I stated that it's going to be the recognition of sperm and the egg. So without recognition, it cannot enact or it cannot take place. Okay. There are several questions asked from slow and fast block to polyspermy. Okay, though I am not explaining the entire concept right now, I would just like to tell you, because if you have an inquisitiveness or interest in studying developmental biology or to attempt these questions, you need to understand the concept of polyspermy. First, I would explain what is polyspermy. Out of several or 20 billion sperms that are deposited, only one sperm would fertilize the secondary oocyte or ova. Remaining ones are rendered ineffective, and therefore a critical mechanism is in place to prevent multiple sperm from fertilizing the egg. This criticality which exists is called as the block to polyspermy. There are two blocks to polyspermy. One is a fast block to polyspermy, another is a slow block to polyspermy. Often questions are asked based on the prevention of polyspermy, that is fast block and slow block.
Sir?
Yes, sir. Tell.
Sir, here the question, the answer is given C and the option given in it is A. And I think answer is C. So like.
No, actually the correct answer is C. Let's go with the Amitabh Bachchan's KBC style of ruling out. First of all, successful fertilization in sea urchin depends on specific interaction between proteins and receptors of sperms and eggs. In view of the egg fall above, which one of the following combination is correct? Okay. Well, I'm afraid what happens is please consider this question as null and void. When you are providing the assignment, I will be repeating the question once again. Right. The reason is I somehow due to typological error only the options have been typed. Actually, it's not the option, just like the previous question. There is a set of combinations A and B, C and D, like that. So right now I will just explain you how to solve the question. Thanks, student, for raising the question. This is a very valid point that you have raised. Even your answer is also not correct to the best of my knowledge. Okay, it's a combination of answers that is going to be. So what I'm trying to convey from fast and slow block to polyspermy is that this section will help you to answer few of the questions. So let's go with the diagrammatic overview, quick diagrammatic overview. Sperm binding and fusion to egg cell membrane, sodium influx. Because of sodium influx, the membrane potential changes. I think I covered membrane potential changes because of influx of sodium within the neurons in my neurophysiology talk. Okay, so this also influx of sodium takes place. Membrane potential change. This change in membrane potential is a prelude for fast block to polyspermy, immediate. In addition, the sperm binding or fusion to egg cell membrane also leads to protein kinases, the kinase stimulation. Kinases in turn will activate the phospholipase C. Phospholipase C, there are two downstream pathways. One is diacylglycerol, DAG, and another is inositol triphosphate. Inositol triphosphate will always mobilize calcium, and diacylglycerol will activate the protein kinase C, PKC. Protein kinase C activation would lead to sodium and hydrogen exchange. Because of the sodium dependent hydrogen exchange, there is an increase in intracellular pH. Once the pH increases, it will stimulate the protein synthesis, the DNA replication, the cytoplasmic movement, and the morphogenetic material. Likewise, the inositol triphosphate production will lead to calcium release. Once calcium is released, it will lead to degradation of cyclin and inactivation of MAP kinase that will restore the mitotic cell cycle. Earlier it was mitotic arrest or Dictyard state. Activation of NAD plus kinase will lead to conversion of NAD plus to NADP plus, lead to membrane biosynthesis. And calcium release is related to the cortical granule exocytosis. Cortical granule exocytosis leads to slow block to polyspermy, and it also leads to formation of hyaline layer. So based on this flow chart, several questions are asked on fast and slow block to polyspermy. Maybe in the practice test that I will give, I will include few of the questions from fast and slow block to polyspermy. Right now, I am not including. My next part is general aspect of cleavage. Cleavage is a series of extremely rapid mitotic division that immediately follows fertilization. During cleavage, enormous volume of zygote cytoplasm is divided into numerous small cells. They are called as blastomeres. By the end of cleavage, the blastomeres have usually formed a sphere. This is called as blastula. I am a bit fast because many of the concepts will be repeated in my succeeding slides. The next is germinal layers amongst the bits and pieces. The ectoderm, yes, alone one slide, germinal layer can give rise to some 10 to 15 questions. And live example I will give you after the portion is over. Germinal layers, the ectoderm gives rise to epidermis, the nervous system, and pigment cells. Before I go further, one small interaction with the audience. If you can unmute or you can use the chat box and tell, someone can tell whether the ectoderm gives rise to epidermis. The question is, hair follicles are there, we know, right? And you have the hairs. My question is, what about the hairs? Are they epidermal or dermal? And what about the hair follicles? Are the hair follicles epidermal or dermal? And by the time, one general knowledge thing I should give in the lecture, you have a muscle. The name of the muscle is erector pili. This muscle contracts, and contraction of this muscle leads to what you call as a goosebumps. In Hindi, we call it as rongte khade ho jana. You get goosebumps. It's because of the sustained state of contraction of this erector pili muscles. Okay. So ectoderm gives rise to the epidermis, nervous system, and pigment cells. Yeah, I did not get any answer. Okay. While your hairs are epidermal in origin, the hair follicles are dermal in origin. The hair follicles are dermal in origin. Okay. The mesoderm generates kidneys, gonads, bones, heart, and blood cells. Okay. The mesoderm generates kidneys, gonads, bones, heart, and blood cells. Yeah, one more CSIR-NET question verbally. Directly you can speak. Right now I am blind to chat box, so someone can unmute your microphone and speak, those who are at least in Zoom. Chat box you can write, but right now I won't be in a position to interact. Okay. Okay, the question is, likewise for endoderm, it forms the lining of digestive tube and the respiratory system. The question is, in humans, can you name the organ system in humans in which all three germinal layers are involved? Can you name the organ system in humans where all three germinal layers are involved? All three germinal layers are involved. Okay. Yes, anyone can use your microphone. Please unmute your microphone and answer. Okay, I'll wait. I will read the chat box in a while. I will go ahead with my talk. And this is the diagrammatic overview. Ectoderm or outer layer will give rise to outer surface, the central nervous system, the neural crest. The mesoderm or the middle layer will give rise to dorsal structures such as the notochord, the paraxial or the bone tissue, the intermediate mesoderm, the lateral mesoderm, and various types of mesoderm. The endoderm, the internal layer of the digestive tube, the pharynx, the respiratory tube, and directly from gastrula you get the germ cells, that is the male and the female pronuclei, the sperm and the egg. Okay. So now one of the question based on germinal layer, bones of the vertebrate embryo are derived from, very simple one. You just get the concepts right, you can answer such type of questions. The options are ectoderm, endoderm, mesoderm, so on and so forth. Can someone unmute your microphone and give the answer?
Mesoderm.
Yes. Option?
C.
Yes, option C is mesoderm. That's right. Okay. So such type of questions can be asked. Just you master the different organs. And what about the other question that I asked, which is the only organ system? Any answer? The correct answer is digestive system. Digestive system is one of the organ system where all three germinal layers are involved. Now I go to gastrulation, just overview, bits and pieces. After the rate of mitotic division has slowed down, the blastomeres undergo dramatic movements wherein they change their position relative to one another. The series of extensive cell rearrangement is called as gastrulation, and the embryo is said to be in gastrula stage. As a result of gastrulation, the embryo contains three germ layers. Just now I saw the ectoderm, the endoderm, and the mesoderm. Just now we saw. Now we will study some of the terms associated with gastrulation, and using these terms we will answer some of the CSIR-NET questions. The first one, invagination. Invagination refers to a sheet of cells called an epithelial cells. The sheet of cells called as epithelial cells. The sheet bends inward. Then ingression, you can see the diagram, invagination. Invagination is just like you are walking, and then all of a sudden there is a lid of the gutter which is open, or a manhole, you fall. That is, you can imagine, you have the sheet of cell which bends inward. It bends inward, that is invagination. Then you have ingression. Ingression is individual cells leave an epithelial sheet and become freely migrating mesenchymal cell, that is ingression. Okay. So individual cells leave the individual cells have left the epithelial sheet, and now see they have left, and now they will be migrating freely migrating mesenchymal cell, that is ingration. Then you have involution, where epithelial sheet rolls inward to form an underlying layer, right? So there is a epithelial sheet, you see it rolls and forms an underlying layer, that is called as the involution. So invagination, ingration, involution. Further, you have epiboly, where a sheet of cells spreads by thinning. It will spread by thinning. Just you have example of childhood chewing gum. You have the entire chewing gum. As you stretch, the chewing gum would become thin. That is thinning of the sheet, that is epiboly. Intercalation, where the rows of cells move between one another, creating an array of cell that is longer in one or more dimension, but it's thinner, that is intercalation. Likewise, you have convergent extension. Convergent as the name suggests, convergence and divergence. The rows of cells intercalate, but the intercalation is highly directional. You can see either it will be in a particular direction, that is convergent extension. So these are the 6 terms. Using the 6 terms, let's see whether we can answer some of the CSIR-NET questions. There it is. Inward movement of an expanding outer layer spreading over the internal surface during gastrulation is termed as, whether it is invagination, ingration, involution, or delamination.
Invagination, A.
Okay. A, invagination. Right now my chat box is active. Earlier I was blind to it. So please give others also chance. But yes, your answer is, what is your answer? Inward movement of an expanding outer layer spreading over the internal surface during gastrulation. Spreading over the internal surface during gastrulation. Would you like to go with some other answer? Okay, by the time you are thinking, let's go ahead. A sheet of cell bends inward, that is invagination, whereas involution is an epithelial sheet rolls inward to form an underlying layer, So the big confusion is between A and C. Okay. So inward movement of an expanding outer layer spreading over the internal surface during gastrulation. The correct answer should be yes. Do you want to try once again, anyone? The correct answer, of course, is C, that is involution. This is the event that takes place during gastrulation. Okay, so the answer is not invagination. The correct answer is involution. Pinkal Shah, yes, I would agree to what you have written, that the cleavage division occurs post-fertilization. That lacks interface. Very true. In fact, I have jumped the gun. Ideally, after fertilization, one should go for cleavage. The sequence in bits on pieces should be the sex determination, after that, the structure of gamete, fertilization, and after fertilization we are supposed to study cleavage. So whatever information Professor Ramchandran sir has given is absolutely correct. He cannot be wrong. The process in which 3 germ layers form is called, well, I don't think this needs any time because this is a pretty sitter. Sometimes such type of questions are also asked. The answer is gastrulation. A hollow ball of cell best describes whether it is blastula, morula, gastrula, or gamete. What's the correct answer? The blastula, the morula, the gastrula, and the gamete. The correct answer is option A, that is blastula. What are cell differentiation and cell specification? Some of the questions are asked based on this. Maybe we'll go to the last segment. The development of differentiated cell types, different in function, cell shape, internal structures, protein and biochemical pathway. This is preceded by commitment to fate. Preceded is before. So what is specification? Specification can differentiate in neutral environment, but reversible in new environment. I will better give the example where you will understand better. Whereas determination is irreversible. Determination is irreversible even in new environment. Okay. Specification can differentiate in neutral environment, but reversible in new environment. Determination is irreversible even in new environment. That's the basic difference between specification and determination. What is autonomous specification? Autonomous specification independent of other cells in the embryo, that is self-differentiating parts. Autonomous specification. Autonomous specification is determined by internal cytoplasmic factors. If cleavage patterns are invariant, then the cell fate would be invariant. It involves mosaic development and is characteristic of invertebrates. These are some of the keywords to solve the answers. Now we go to conditional specification or non-autonomous specification. It depends on conditions, that is cell interactions. More variable, that is variations in which the cells are assigned which forms. Non-autonomous or conditional specification has capacity to regulate. It's the characteristic of vertebrates and some of the invertebrates. The third category is syncytial specification. Syncytial specification, the interactions between regions of cells before cellularization has taken place. The cells have no specific cell fate before cellularization. However, after cellularization, mostly conditional specification takes place. And the examples are insects. Of course, students, let me tell you, this topic specification itself can take 2 hours depending on what depth it is being taught. But right now, I'm just giving an overview because we are into a crash course. And based on specification, we have one experiment, and this is a very famous experiment of Rox, the famous embryologist. What Rock's experiment is all about, it's a defect experiment. What Rock's did, he took frog embryos, you can see, fertilized frog egg, and using a hot needle, he just made, he pricked using a hot needle one half of the embryo, okay, at a 2-cell stage, the blastocyst stage. Thereafter, he allowed the developmental process to complete. What he found that part of the embryo which was pricked with a hot needle had dead tissue, whereas the other half had living tissue. In other words, Rock's got a half embryo, okay, half embryo at the neurula stage. This, it leaves us a question mark whether it supports autonomous specification or not, okay. Whereas we have other experiment, okay, tunicate specification is mostly autonomous, and there it you have tunicate embryo. One of the experiment that was performed, it was an isolation experiment. Separate 8-cell stage quadrants are there. There is quadrants, separate 8-cell stage quadrants, that is 4 plus 4. Each forms what it would have formed and does so autonomously. Even if whatever each quadrant was to form, exactly that quadrant forms the exact structures. So this is an example in tunicate embryo of autonomous specification. Okay, autonomous specification. Some of the questions based on specification, just to give you an idea that how this topic questions are asked. If you remove a set of cells from an early embryo and observe that the adult organism lacks the structure that would have produced from these cells, therefore the organism seems to have undergone which of the following option: autonomous specification, conditional specification, morphogenic specification, or syncytial specification. You remove the set of cells from the early embryo and observe that the adult organism lacks that structure that would have produced from the cells. Therefore, the organism seems to undergo what? Autonomous, conditional, morphogenic, or syncytial. The correct answer is option A, autonomous specification. Okay. When an 8-cell, directly from what we studied just now, few minutes before, when 8-cell embryo of tunicates is separated into 4 blastomere pairs and allowed to grow independently in culture medium, then each blastomere pair can form most of the cell types. However, the cell for nervous system are not developed. Remember, it's not developed. The following statements are formed from the above observations, June 2010. These are the statements. First, nervous system development demonstrated autonomous specification. Yes. The other tissue types are formed due to conditional specification. All the tissue types except nervous tissue that demonstrated autonomous specification. nervous system development demonstrated conditional specification. The question is the correct combinations of statement. You have to find which is the correct combination of statement. Each of the statements are correct. Which two correct combination would held in place? For this, you have to understand the difference between autonomous and conditional or non-autonomous specification. Okay, since we are on the borderline of time, what I would do, I would just give the answer. The correct answer is option C, that is both C and D. C stands for all the tissue types except nervous system that demonstrated autonomous specification, because nervous system, even without that, it formed nervous system. And D, nervous system development demonstrated conditional specification. Correct. So quite obvious, both complement each other. Since nervous system is behaving in an abrupt fashion, that demonstrated autonomous specification, right? Except nervous system, all other demonstrated autonomous specification, whereas nervous system demonstrated conditional specification. So such type of questions are asked from the specification part. It's very important in developmental biology. Now we have induction and competence. Finally, I could get the 3 text messages which Pinkal Shah has written. I have gone through all the questions, right? The genetic information in male gamete that fuse during fertilization passed in every cell, but already the meiotic arrest in oocyte has taken place. Okay, despite that, Pinkal, at this stage, I cannot give you one-liner answer, because the question which you have asked might take there are multiple paradigms to answer your question. You have differential gene expression, you have egg activation, you have capacitation. So multiple mechanisms have to be addressed, only then I can answer your question. Right now, directly, it's difficult for me to answer your question, though I have understood your question very perfectly, that despite being the post-fertilization lacks interphase, okay, so how is the information passed on to cleavage? Okay, so that's your question. I have understood your question, but at the moment it's difficult to answer in 1 or 2 lines. That is a separate lecture of cleavage that has to be understood, and how cleavage is linked with fertilization. Okay, so induction, which at close range between 2 or more cells. For induction, we have an inducer, the tissue that produces a signal, a responder, the tissue that is being induced or target tissue, and finally, competence, the ability of a cell or tissue to respond to a specific inductive signal. Okay. So you have the flow chart to explain, and this might be the last concept, and then I will wrap up. The inducer, the tissue region secreting a morphogen, autonomously deforming tissue region. You have a signal. It can be a diffusible morphogen or a mechanical force. This induction of the responder, and for the inducer to signal the induction of the responder, you need competence of the responder. The competence of the responder is very important, right? Just like the English proverb, you can take the horse to the shore, but you cannot force it to drink. Similarly, the competence of the responder is the one which will signal the inducer for induction of the responder. Based on this concept, one quick question to wrap up. The ability of the cells to achieve their respective fates by interacting with other cells is known as the ability of the cell to achieve their respective fate by interacting with other cells is known as autonomous specification, conditional specification, induction, and competence. What is the correct answer? Just now we discussed about nervous system, so things might be cell to cell communication is important in development of an organism. The ability of the cells to respond to specific inductive signal is called as, from CSIR 2015, regional specificity of induction, competence, juxtacrine signaling, or instructive interaction. Perfect, bang on target, most of you. It's competence. Just now I showed the flow chart. Okay. I would have been delighted to answer Pinkle's query because I had taken a short brief of, okay, with respect to the development of an organism, autonomous specification would result in what type of development? Regulative development, mosaic development, syncytial development, or definitive development? Arpit Soni and Nirali Tandal has got C, that's right. But is it right? In fact, it was there already in my slide. Okay. The answer is option B, the mosaic development. Okay. The mosaic development is the right answer. Nirali might have to go to my YouTube link for getting her answer because I have a separate video I will just share with her after this talk of her 3 long text messages. I could get her questions correct. Maybe after listening, she will be cleared with her doubts. So due to time constraint, I am, as such, the title of the talk was bits and pieces. So one of the piece I am leaving for this lecture, cleavage. Okay. So there we had the link between the fertilization and cleavage. The various types of cleavage, again questions are asked. Types of yolk, egg, the types of egg based on yolk distribution questions are asked. Okay. So these questions, of course, I will give you in your practice session. So like every time, those behind me, because alone it's not possible with all the working load of the universities. Megha madam or my student since last 12 years, Megha Dave, for compilation of the question bank and for comprehensive reviewing of the questions. Ansuya madam and Dr. Darshi Bakshi and Elizabeth, Dr. Elizabeth from the host side for providing the platform. Last but not the least, thank for your kind attention. While I convey thanks to everyone, two note I would like to add before I hand over the microphone to the organizing team. I have designed the thank you slide as a perfect means of developmental biology, but then students, sometimes we have the compulsion that we are going in an overview fashion because of the time constraint and it is a crash course. But if you ask me the question, right, like my mentor, Professor A.V. Ramchandran, or any other experts, I am also delighted in giving in-depth topic of a particular topic, just like the T-cell biology or your neurophysiology, rather than going in the bits and pieces. But then the subject is such that in Gujarat, most of the biotech and micro student, they are not exposed to this subject. Every university developmental biology, for Navrachnites, you are lucky you have this subject, but others don't have. So therefore I thought that rather than leaving everything in option, at least I made it a small attempt to introduce you to the subject and with some bits and pieces. Maybe you can keep waiting for my next videos exclusive on cleavage, on fertilization, and on Drosophila axis formation. They are some of the frontier areas of developmental biology where questions are asked, and the entire lecture is spanned in 2 hours for a particular topic rather than moving around with topics. With this note, I hand over the proceedings to the organizing committee. And for Pinkle, of course, our interaction through email would continue. My email is given in the very first slide of my lecture. You can refer back. Over to you, Elizabeth, madam.
Yeah, thank you, sir, for a very informative session. Big topic you had tried to concise in the 2 hours, and I hope that they could understand the difficult things which you made it in a very simple way, and I hope it has helped them in their preparation for the NET exam. And of course, all the questions can be, will forward it to Ragdim, sir, if you have, or you can directly even talk to him. And of course, we will be sharing the PPT also with you as well, but after the workshop is, so that you all can attend all the workshops every day. Okay, thank you so much, Ragdim, sir, for sparing your valuable time and for making a lot of things very simple for the students to comprehend. Thank you so much for all the efforts that you have made for all the 3 sessions that you have taken. And students, we stop for this session, and we meet at 2 o'clock for plant physiology. So see you at 2 o'clock today. Thank you all so much.